Neil Spooner, the chair of the Patient-Centric Sampling Interest Group, and I'd like to welcome you to the part 2 of the PCSIG workshop, A New Era in Clinical Diagnostics, the Power of Self-Sampling. So this is part 2. In part 1, we very much talked about what the challenges are for the more widespread implementation of patient-centric sampling for clinical diagnostics. Today, we're going to very much focus on what the solutions are to those challenges and what the next steps might be. So I've just got a few slides to go through to introduce before we start the discussion. Obviously, we want to thank all of the PCSIG partners and sponsors, without who we couldn't be putting on this event today. We couldn't be putting on our webinars and all the other activities we have ongoing. And thank you to all our members that support our activities, that give up their time for free. So today, just an outline of what we've got today. We want to very much focus on solutions. So we're not going to be looking at challenges anymore. And because we've just got an hour, we can't focus on all the challenges. We've prioritized 3 of them that we're going to talk through this afternoon. But the idea is that we talk through these solutions, but also we want to spend the last few minutes of the meeting talking about further future activities for this group. We have to remember that a similar activity, similar workshop to this is ongoing in North America starting next week, and we'll be wanting to find out what the output from their workshop is as well before we select too many ongoing activities. But as we're going through this is not just, remember, this is not just a discussion for today. We're looking, how can we deliver real and long-term solutions to these challenges? And that will mean a number of activities needing to be agreed, and hopefully members of our PCSIG taking part in those activities. So before we get any further, I'll just close off these slides for a second and let everyone introduce themselves. So if we can start with Vincent, could you introduce yourself, please?
Of course, Neil. So hello, everyone. My name is Vincent. I'm affiliated with Greiner Bio1, where I'm a global market manager. I'm responsible for our Capillary portfolio and the pipeline for that. And next to this, I'm also affiliated with the Leiden University, where I'm doing my PhD in microsampling. So nice to be here.
Thank you, Vincent. John?
Hi, everyone. My name is John Fabule, and I represent the patient involvement and collaboration community. I'm a physician by background, and I specialize in pharmaceutical medicine. Great to be here with you.
Thank you, John. Joseph, introduce yourself, please.
Thanks, Neil. Good morning, good afternoon, good evening. Thanks so much for joining. My name is Joseph Burt. I'm part of the MHRA, the UK regulator for medicines, medical device and blood components for transfusion. Thank you for joining.
Thank you, Joseph. Sharon?
Hello, thanks everyone for joining. I am the current microsampling chair for the IFCC, and I'm a pathologist and have been a medical director for my career at Stanford University School of Medicine in the United States in California.
Thank you, Sharon. Tim?
Hi, my name is Tim Woolley. I'm the Chief Scientific Officer at Untitled Diagnostics. We specialize in capillary blood testing and sample stability.
Thank you, Tim. And last but not least, Rachel.
Hi, everyone. My name's Rachel Cowling. I'm a consultant scientist and director of newborn screening at Synnovis, and we're the pathology providers to Guy's, St Thomas's and King's in London.
Thank you, Rachel. So just to take you through, for those that are online, thank you for joining today. Please use the chat function to submit your questions and suggestions for solutions and further activities. The workshop is being recorded and will be available on demand through the PCSIG website after the event. So following part 1, we had a number of challenges around the patient, sample quality, labs, clinical, logistics, data management, economics, and regulatory. And we've decided that we only have enough time to cover a few of these today. We're going to ignore the rest. They're going to go into whatever output comes from this, and hopefully, if there's working groups come from this event, we'll carry that forward. So, no further ado, I'd like us to start off with a patient-centric sampling interest group, and this is all about patient-centric sampling. I'd like to talk about the patient to begin with. So I'll just close that down. Vincent, you were talking earlier about what you think is the importance of knowing what the patient wants, whether the patient has collected a good quality sample, etc. I just wondered whether you'd like to go into that a little bit more, please.
Mm-hmm. Yeah. What I think when, of course, we were talking about the challenges is that the patient needs to collect the sample, him or herself. And I think what's very important when we are implementing solutions like microsampling is that we focus on what's actually the need of the patient. How can the patient properly collect the sample? So I'm thinking of how do you properly instruct the patient? Because it has also been shown that if you do not properly instruct your patient, you end up with underfilled tubes, for example, or lower quality. So I'm thinking of, for example, digital platform that should always come along with an implementation of microsampling where a patient can follow a video or can be accessing a professional that can help guide them, for example. But also, I think especially in pre-analytics, it's known for its errors, of course. It's the most critical phase. And so when you move it from a healthcare professional setting more and more towards a decentralized setting, I think also the patient identification part is very important. And maybe we can take away of some of the other industries examples, like the KYC, know your customer. Maybe there can also be similar approaches to also verifying the patient in this case. Just some ideas and solutions for also to tailor it to the patient.
Yeah. No, I completely agree with you, Vincent. Can I go next? I just need to.
Absolutely, John.
Yeah. So I think when I look at it from a patient perspective, like myself, I think there's a human side of it and there's a scientific side of it. Because as a patient, you have to have confidence in a test or diagnostic before you use it. Because if you don't have confidence and trust in it, you're just not going to end up using it. And how can we ensure that patients have that confidence in that test? So patients are worrying, but for example, you're thinking, well, if I do this test, will I end up repeating it again? Will it be trusted enough to actually be fit for purpose, like the standard test that were validated? So these are some of the questions that come up in patients' mind when we are thinking about confidence as a barrier to patient-centric sampling. So when I look at it in that manner, I think what is important for us to be able to ask is, what exactly would the patient need in terms to have confidence to actually use a test? And if we actually factor that into maybe a small working group or discussions, when we have discussions like this, then we can actually take that away and the patient can contribute usefully to say, well, for me to have confidence in that test or that diagnosis, these are the list of things I need to actually see. I need to make sure that it's actually validated to actually do it. I need to make sure that, when I do it, I don't have to repeat it again. And once we get some of these patients' voice and opinion and comments into a working group, I think it will actually make for a better adoption and better uptake of patient-centric sampling from a patient perspective, at least.
So, John, are you suggesting that the solution is for each use case for people to engage with patients in their local area to do this? Is that what you're talking about?
Yes, exactly. Because, it's not about the broader PCS as well. Because as a patient, you're looking at that test you have been asked to do now. So for which patient, for which decision, for which pathway, But because that's the question I'm being asked. So if I do this particular test now, do I really trust that it's going to actually be fit for purpose? And so I think we need to concentrate on that as opposed to the general. I mean, general is still important, but the specifics that the patient is going through at that time will be very important as well. So patient input into NAR, I think, is critical at the very early stages.
So can I butt in there, Neil? Sorry, can I just?
Yeah, absolutely, Tim.
So when we did our IFU, we've got three options. We've got a hard copy, we've got a YouTube thing, and we also offer online training, for instance, live online training, or we've had groups of people that we've trained. But when we did our IFU, we involved part of that process to get it CA marked was to involve the patient cohort we were aiming at. Now, to be fair, ours is probably a different cohort than most, because we're a remote lab, therefore we don't have a hospital attached to us. But most of our people are wellness, for instance. But we involved those individuals, and we went through our IFU to make sure that any of the things that we took for granted, they didn't, and we built that into the IFU.
Yeah, great.
Perhaps, Neil, something like a recommendation based on also the experience that Tim is now sharing. A recommendation of how the patient instructions should be formulated in the hard copy or the video. There's probably some lessons learned, and that can be created as a guideline to help other people implementing microsampling solutions.
Yeah, I like that.
Yeah, and one of the other things we can also think about is short summaries as an output from that kind of working group. So where the patient and the clinicians and lab actually look at that pathway and look at where patient voice is most valuable in that pathway, and you can actually have a minimum kind of recommendation to say these are the summaries that we came out as a working group, and this is what patient voice will be most useful. And it doesn't need to be a long list; it can just be an executive kind of summary.
Okay. Just got something coming in here from Karen. How do we help the large percent of older patients with long-term conditions that require monitoring that capillary sampling can be as reliable and more accessible than venipuncture? Our older patients still like to come to clinic for bloods. Just any comments around that? Thank you, Karen.
Only that, so we had a discussion with a group of doctors not so long ago, and they said that they still wanted to see patients. So therefore, they were going to do self-collection or aided collection in some cases, because they were going to actually do the capillary sampling for the patient there and then. So there's that opportunity where all you're doing really is removing the phlebotomy. It's still collected by a medical professional. And of course, the other thing as well is once they've seen it done a few times, you're actually providing training to the individual should they decide eventually to want to do this at home. And after they've seen it 3 or 4, 5 times, they may go, actually, you know what, the next time I can do this at home.
Yeah, and I think based upon what we've seen in pharmaceutical clinical trials, we know that the success of collecting a good quality sample is improved immeasurably if the first time it's done in a clinical setting with oversight, and then people are happy to go home and do it themselves. But people just receiving a kit through the post just doesn't work really. It's likely to get a high failure rate. So, you know, one of the solutions we seem to be coming up with here is to build a PCSIG based recommendation about what instructions should look like, what training should look like, and how to include the patient in that discussion. Is that a fair enough thing to say that we seem to be coming out with that as a potential solution?
And I would add, Neil, that there needs to be learnings taken and incorporated into iterations of the product as more and more experience is taken and data is accepted in the use of the product. So you might have a first iteration of product for microsampling and sample taking, but it's then used, and over time you get to learn a little bit more about the product. So the uses of that product, that sampling, is then incorporated into the learnings, into the instructions for use, in the way that maybe the device is used, and then incorporated in the improvements of that product over the longer term.
Yeah.
And one other thing, if I may actually discuss this in several other forums before, is that the lab and the patient use different sort of language. Lab speaking scientific language, and patient just speaking ordinary terms. So how do we ensure that these two actually are able to understand themselves? So a lexicon sort of so is what we've actually discussed before, where both actually understand, they have to come to the same understanding, especially when it comes to use of some of these scientific terminology and jargons. And patients working with lab on that would actually help, in my view.
Yeah. So, do we have any other thoughts about solutions around the patient at all?
I would just like to say that when I'm listening here, I'm hearing about sort of two different settings. One is when a manufacturer is going for a CE mark or an FDA approval, there's a high level of patient engagement required, obviously, and that involves one sort of scenario. But then later, when the CE mark or the FDA mark approval has been achieved, then we're talking about clinical settings, and we're talking about the lab. And the model that's the best sort of proven is the diabetic model, where people have been self-sampling for many decades. And that's typically involves instruction in the clinical setting, where they're seeing the patient, maybe a nurse educator or some other type of individual, but it's not really happening in the lab as much. And I think that the clinical setting is sort of a better model than putting all of that instruction on the lab, which really isn't, it's a lot of labor, and it may be a bit burdensome.
Yeah. Just got an interesting comment from Morris here. Indeed, last week on the Dutch clinical chemistry meeting, the discussion was that IFCC clinical chemists should have more saying in setting up standards for self-testing home sampling lab tests. So what do we think about that comment from Morris?
I would agree, Neil. There is already a standard, an international standard for this type of work. It's ISO standard 62366, and that looks at human factors engineering. And believe it or not, we actually, as regulators, look at that very, very deeply. So human factors is a very important part, especially when you're considering how your device is used in a lay setting, a non-healthcare setting, and the applicability of that device for use in that setting. So that standard already exists, Maurice, but you're absolutely right. There needs to be greater adoption of these standards, including the one I just mentioned, 62366, which is a very well understood and global standard that should be adopted, and many regulators look at that standard as a benchmark.
Okay,
Perhaps it's something we could do together with the PCSIG and IFCC, is to formulate a recommendation paper or a recommendation guidelines on trying to apply all all these regulations into a microsampling? Could be a suggestion.
So the patient testing group that we have at the LabMed UK, the PACS group, which is the Patient-Centric Testing and Sampling Interest Group, have written a paper on the recommendations for validation of capillary blood sampling in laboratories. And that does include the whole process validation from patient collection all the way through to reporting. And that's currently being reviewed right now. I think it's gone to the annals of clinical biochemistry.
Great.
Yeah, great.
So just comments under here that this falls under ISO 15189 as well, diagnostic services. So that's coming from ANIC, and that's working quite well. She's saying, let's not make it more complicated than it is. Don't know whether anyone had to comment on that at all.
Yeah, I do, Neil. So just let's be very clear what that is. 15189 is the standard for medical laboratories. So that is how you operate the laboratory in an accredited way. So here in the United Kingdom, we have UCAS. Similarly, in the Netherlands and the rest of the EU, there's the accreditation services that you would get needing to get accreditation for. That's standard, but it does not negate the fact that your physical device, your self-sampling device, still needs to consider the human factors elements, and that's what I mentioned in the other standard 62366-1 that needs to be followed.
Okay, thank you, Joseph. Are we naturally moving on to some of the discussions around challenges for the labs here? Is there anything else to cover on the actual patient side of things? Okay, so we'll move on to some of the lab challenges. And I know one of the concerns has been around integrating samples from patient-centric sampling approaches into current workflows in diagnostic laboratories. So what could be a potential solution in that area? How could we go about overcoming that issue, and who should we be working with? Manufacturers?
Yeah, so you've got the three analytical phases, obviously pre-analytics, analytics, and post-analytics. So we've done a little bit with the pre-analytics, but that's just getting the sample to the lab, and there's a few other things in there that we could discuss at length, things like, whether it's postal or whether or not it's dropped off at a GP surgery, and do you have to include sample stability and things like that. So, but I won't discuss that. I'll leave that to one side. But once the sample gets to the lab, this is basically a pediatric sample. It's a sample tube, and you can process it in a similar way. So people who've got experience in doing pediatric tubes know that it's not as simple as the venous tubes, but likewise, it's that same process. It's just a smaller sample to deal with. So we have a setup that deals with lots of it, because we do lots and lots and lots of that type of testing, and we've had to. We're not a massive lab, and we don't have lots and lots of big equipment, the big pre-analytics and post-analytics, but we have figured out how we can deal with those samples. And actually, once you get it past the initial opening up stage, it fits into the process quite simply.
So if you're saying it fits into the process quite simply, Tim, do we need to find a way of sharing that more widely? If you're saying it's simple, but other people are saying it's a challenge, there's obviously a mismatch here. So do we need to get better?
During COVID, we were doing about 9,000 capillary samples a day. So we learned a massive amount from that. So we find it now simple because we're only doing 1,500 a day. Therefore, it's for us it is quite simple. But what we do is once we've got the sample and opened up and go through all the patient labeling issues that people have talked about, which again, overcoming that's not too difficult. Once the sample's centrifuged, we then aliquot it. All right, and that's probably our biggest problem, is that we can't put the primary tube on the analyzer. The analyzers just don't like it. And it's fine if you've got a disposable pipette tip, all right? If you're going to put your chemistry analyzer with a fixed pipette tip into a little gel tube, you are going to be down most of the day. So we aliquot ours into a secondary tube, and then you have the point of having to label those aliquoted tubes. That's the main problem we have. But once it's been aliquoted, it's good on the analyzer. After that, it's like any other sample, for us at least.
So,
oh, sorry, Neil.
No, you go, Vincent.
Yeah, Tim, I was just wondering, indeed, you're aliquoting the samples. So I know also that sometimes primary tubes are used directly on the analyzer, but I'm just wondering, in terms of solutions, do you also see potential changes to either the analyzer or the tubes to make sure they could get rid of that aliquoting step?
Yeah, so that's really interesting. So we've just been looking at there was massive amounts of effort put into the collection technology. So we used to use finger prick, and now everyone's going to transdermal, and transdermal has been a game changer for this technology. All right, it's really great. But the pediatric tubes that are in use, I've actually been out there for, well, I think I've been in it doing since the 90s, and I think they were out then. All right, so but they haven't moved, they haven't really changed. And that's part of our problem, is that all the attention's been focused on the patient collection part, which is brilliant, fantastic. But now we're dealing with legacy tubes, if you like, and we just can't get them on our analyzers. When it was doing COVID antibodies, we use the Roche. It's a disposable pipette tip. So if it goes into the gel, the analyzer just goes, it's insufficient, it's clotted, bins it. And that's fine for our immunochemistry, but our chemistry analyzer's got a fixed probe, and we are going to get gel in that probe more or less within the first 5 or 6 samples if we put it on the analyzer with primary tubes. So we have to aliquot. All right. I mean, aliquoting gives you a couple of benefits, in that you see the quality sample before it goes on. So if it's insufficient, you don't need to carry on analyzing. It's just insufficient. But we do analyze. That's what we have to do. And automating that's a problem. All right. So that's where we suffer, because we're not a massive lab. But other people in the US have already conquered this. They've got a device that uses something called LabEye, which is monitoring the sample amount, and they've conquered this. But they put a massive amount of money and a massive amount of effort into that. We're a little bit behind that in terms of the pre-analytics.
Yeah, I was thinking also in terms of solutions, there's also indeed automated aliquoting instruments. But so now that the instrumentation companies can also work with much less volume, and also, as we discussed last time, is that maybe we should also pivot a lab or change a lab to, I think it was a comment of you, Rachel, to the need of microsampling. And it can also be the tube, of course. And so maybe I'm just wondering, Tim, if it makes sense to eliminate the aliquoting step, to have a different tube design that has a bigger column of serum or plasma in that case.
Yeah, I mean, if they could do that, that'd be fantastic. Or if, I'm talking with what we have right now, and we've come up with solutions to solve some of our common problems. But yeah, I mean, we're using old tubes, or not old tubes, but we're using old style tubes. So if someone came up with a newer tube that maybe removed the gel from the problem and just left you with the serum that can go labeled and go straight on the analyzer, fantastic.
Yeah, could I just chip in? It's more of a comment. I think for me, the key thing is to sort of acknowledge that it's a separate process, and then what happens then depends on your laboratory and your setup, and obviously that's different for everyone. It also obviously depends on the device that you're using. But I think as somebody who works in a sort of an automated, well, I don't personally work in an automated clinical chemistry lab, but that's sort of the thing we're talking about. That's the thing, is it is a separate sample process. I think that's the key point to remember, because the question then, if you can acknowledge that, you're not trying to force it to fit with your existing process, which is very different, where a lot of the problems certainly in the past have stemmed from, I think.
So is the solution here to put a team together to produce some sort of best practice document? Would that be useful or is that already out there? We just need to draw people's attention to it more?
I think certainly getting the manufacturers involved, that's be fantastic, because that would could probably come up with some fantastic ideas and solve some of the problems that we've got straight away. So that would be fantastic. The recommendations paper we spoke about does cover some of these areas, so that will be hopefully coming out soon, and that will help a lot as well. Give a baseline, but I think getting the manufacturers involved and looking at the process is like everything, is critical.
Yeah, and Neil, I would agree exactly what Tim just said, in that really the fundamentals here is not just to act in isolation, but to work integratedly with the assay manufacturers, the instrumentation manufacturers, and the automation manufacturers who help actually operate the lab itself. So you may have a process of sample collection coming in, but then you still need to do something with that sample. And as Tim said, it's actually being able to work with those manufacturers to help maybe simplify the lab process as you are bringing in those samples into laboratory processes.
So just moving the conversation on a little bit, and there's been some suggestions. So people who are making suggestions in the chat, please bear in mind that will all be captured and will go into the output we have. We just only have enough time to read and comment on so many of them. So Karen, your fridge racks and things like that sounds like a great idea and a great solution. So thank you for that. There also seems to be a lot of discussion about reference ranges, established reference ranges, and the new sample types, capillary blood, dried blood, whatever, not fitting into those reference ranges. There's quite a lot of chat going on amongst our audience today about that. Anyone want to talk about that at all?
I suppose I can chip in again if you wish.
Yeah.
So we've done the comparability studies. So our lab, we do the typical everyday chemistry, immunochemistry analysis. So we wouldn't be reformulating reference ranges. If we found that a venous and capillary sample weren't comparable, we wouldn't just offer it. That would be it for us. Rechanging reference ranges for us would be a no-go. I'm not saying that wouldn't be for other people. Larger laboratories that've got specific patient cohorts, but we're using the standard reference ranges that have come from the manufacturers or from group bodies or things like that. So we do comparability studies, and if it's comparable, then we accept it.
So I guess the question comes is, how useful is one lab's comparability study to another lab's comparability study? How much can that be tracked across?
Well, there's a lot more papers being published recently on the comparability of venous blood and capillary blood. I mean, there's 4 or 5 years ago, there were hardly any, or specific instances. Now there are groups all over the UK and Europe have published widely on the common ones, and the list of assays that have been proven to be comparable is forever growing, and a lot of those also include whole blood stability, 3-4 days, different temperatures, so that data is growing forever. What I would say is I would recommend rather that each lab do their own validation rather than rely on published data. It gives you an idea that's possible. So, for instance, if someone says it doesn't work, then it might not be suitable for you. But if someone's already proven it does work, then you've just got to do the validation to make sure it works in your process on your analyzer, using your tubes, using your postal process, and so on and so forth.
I think it's a really important point, because I think you need to be mindful that you may be using a different method to the lab you're trying to compare with, and even if you're using the same method, depending on the equipment you're using, it's possible that you could be calibrating to either different manufacturers' calibrators or in-house or whatever. So there's standardization and harmonization issues to be aware of. I think for some of the more esoteric analytes, or those done less frequently, there's probably a greater degree of flexibility. But if you're talking about potassium, for example, you would need to be absolutely spot on, and you need to do a really comprehensive verification, validation, I think, to be sure that you knew what you were reporting and where it sat against what was being used routinely in, or what's being reported by your local laboratory.
Mhm.
And then again, it's sort of what's that result going to be used for? You can always have comments that go out with reports, caveats, explanations, and it does ultimately depend what you're going to do with the result.
In that sense, I think there's a need for clinical, the clinician's input as well. How comparable does it need to be for the use it's being, for what they're asking from the test?
Yeah, I think that's a good point, Sharon. And obviously, I work in a hospital-based laboratory, so I've got a very different perspective. If you're using microsampling and monitoring, or not monitoring, but looking at different parameters in healthy people, it's a different kettle of fish. So I think it comes back to what you're using it for, doesn't it?
Yeah.
Yes.
So are there documents out there that give clarification of validation versus qualification? Because I know, Tim, you said each lab doing its own validation. Is that the case, or do you just need to qualify and not fully reproduce everything that someone else has done, or do you need to fully reproduce what someone else has done?
Personally, I would do my own whole process validation. We treat it very similar to, for instance, doing a serum to plasma verification, for instance. So, you're wanting to put assays onto a plasma tube. So we do a like-for-like analysis that's based very similar on that. And in fact, it's actually EP35 that we use as our guide. So it is a validation, but we would do our own. We wouldn't rely on somebody else's work, only because this is quite new. And like Rachel says, we might have a slightly different analyzer, slightly different process, slightly different assay. Rachel brought up last week about creatinine. One of the papers said that creatinine wasn't comparable. They actually didn't mention the fact that it was a Jaffa method. They didn't mention that within the paper. And we knew it was, but we went and did again. But we found that the enzymatic creatinine was the one that was right. But some of the papers that do it might not be using exactly the same assay, might not be using exactly the same process, same tubes. So I would recommend that people do a sensible validation process.
So just building on that, Geaghan made a really good suggestion here. Do you see opportunities for labs to work more collaboratively in this area? And are there potential solutions to establish these correlations jointly?
Yeah, I can chip in there. I think there's always potential, and collaboration is always very powerful. But again, I think you would all need to be either doing exactly the same thing, or you'd need to be very clear what the differences were, so you don't tie yourself up in knots, which is surprisingly easy to do.
There was a question a few years ago that I went to a meeting, and they discussed this within NHS networks, where one lab had done the validation project, but all the labs within that network use the same analyzers, use the same process. So did they need to go and do the validation for each platform? And although that wasn't answered, that was a very good point. The fact that network is working closely together, same system, same reports, same process. And would you need to do a full validation for each platform, for each lab? So, yeah, I think working collaborative would be a good idea.
I think going back to your question, Neil, ISO 15189 is quite clear whether you need to do a verification or a validation. Yeah. And then obviously, as Tim indicated, you have got personal choice as well. But I think the absolute minimum is a verification. And if you're verifying something, you're normally verifying the manufacturer's claims. And the assumption is that you are verifying and you are doing exactly what the manufacturer set out in their method. If the slightest change in that, be it a sample type or even a step that you've changed, would mean that you'd have to do a validation. And then obviously I'm obviously talking about clinical laboratories and clinical assays. So it's slightly different if you're offering a different type of test. But for clinical labs, I do think it's quite black and white, certainly what the minimum requirement and expectation would be.
Okay.
Rachel, I agree on that.
Okay, so
I think maybe one thing I want to add here is that also, as Tim mentioned, that currently there's a lot of papers being published also around, the comparison, the analyte comparability. And I think there's a very good base now where you can see, for example, clinical chemistry analytes where give or take 2/3 of the analytes are possible, comparable to venous. And there will always be, in my opinion, analytes that will be more difficult for comparability. For example, potassium, because I saw Nick and Maurice also mentioning here in the comments or in the chat, is that there's in certain capillary sampling methods, there's also shear forces on the erythrocyte. So there's a certain hemolysis degree introduced to the sample, and therefore it may be difficult to have a new reference range, for example, for these more difficult hemolysis-prone analytes, because the effect on how this occurs is very imprecise. So there's a lot of variability. So if you want to create a reference range, then it will be it's not possible because it's a completely random effect sometimes. So I think for some, especially when we're thinking about microsampling, right now with the evidence base, we can look at approaches that can work with the analytes that are comparable, and agree that for the moment, there are some analytes that are not possible. I think that's also an important reality to emphasize.
Yeah. John, were you saying something there?
No.
Okay. So am I hearing that potentially the future action here is maybe putting together some sort of activity with the manufacturers of the platforms, the automations, the actual assays to bottom some of these things out? Are there any other actions coming out of this that build on the solutions that are being offered?
I think that's about right. You've got to remember that also some manufacturers have already started this process. So Roche, for instance, recently released a claim extension on some of their assays that included capillary blood into the IFUs. So that process has started with some manufacturers, so they realize the importance of it. So that then goes from a validation to a verification. And I imagine over the next couple of years, most manufacturers will be adding on this.
But you think if maybe we were to help to get them in a room together, that might help that or hinder it?
No, I think it would help.
And if we were to do that kind of thing, would that be best done under, I'm just thinking, Sharon, would that be best done under your auspices rather than the PCSIG?
I think that there'd be a lot of interest in that, yes.
And, working with national and international societies, obviously facilitated by PCSIG, but maybe we're not the best people to take the lead on this, just thinking that through. Do you think there would be the will in LabMed, IPCC, other organizations in putting this together, maybe across multiple national and international organizations?
Absolutely.
This suggestion made a meeting session at an IFCC Congress. Industry is always there. It's a good suggestion, Maurice. Okay. Some ideas for next steps. So the last thing wanting to cover that we agreed as a team to just go through a little bit before we close today is the economic side of things. So difficulties in getting reimbursement, difficulties in building good cost benefit models to actually show to people we can do this. It's one thing to have the patients willing to do it and the labs willing to do it, but if no one's willing to pay, we don't get anywhere. And we're increasingly hearing of that. So what are some solutions that are out there to maybe making that a little bit more easy for people?
I was just thinking that maybe we could use something similar to what we just discussed now in terms of inviting some of those payers to some of the discussions to actually say, if they're going to invest in PCS, what does that look like? What do they need to see as evidence? And getting some of those payers in might be a good idea, and it might be at an international level as well. It might be at the PCSIG level, it might be at Sharon's level. So, but getting their input, I think, will be critical.
Yeah, I like that, John. Figuring that out, because obviously there's a lot of different payers all across Europe, but you can't try. Well, yeah, we're not even thinking about North America next week. And it may be that kind of discussion with payers needs to be separate discussion in different regions of the world, depending on, how public health is delivered. I don't know.
Right.
So Karen had just made a good comment about the early thing. We were beginning this type of discussion with manufacturers with LabMed UK in September. Watch this space. So yes, LabMed are going to be running an event, and thank you for reminding me of that, Karen. She's also got just capturing economic benefits around travel time to clinic, time off work, decreased fuel for travel are all useful indicators for adoption, but how do we get this data? And I think that's a really interesting thing. There's been some great publications out there. There was one a few years ago in PLOS ONE from the Netherlands that really dug down into the depths of it. Time off work, public transport costs, time of kids off school, and managed to put a euro amount against that, which was really interesting. Can we build tools that help people do that? I'm hearing crickets.
Ideally, this is of course the data that you would need, and I think the reason why there are so few studies at the moment is that it's really difficult to quantify this. But I would also agree that, of course, when building the business case around microsampling, the way costs are distributed is going to change. And I think the only proper and good approach to it is actually doing a good proper study on it, where you compare multiple components and also maybe some outcomes like patient satisfaction. I know in some healthcare systems, patient satisfaction is also an important measure for reimbursement, for example. It's more on health economics, of course, and I think.
I agree with you, Vincent. I was just wondering whether working with patient groups might actually, who are already interested in this area, might actually be a way to get some of that data. I know patient involvement and collaborative community and some other patient groups might be interested in actually working with their with patients in our community to help get that data. So it's something to think about, whether working with those patient groups might actually help.
Yeah, and people, Anik making a good comment here. Maybe home sampling does not need to be cheaper per se, is adding other things and solving labor shortages in the clinic. And it's just, figuring out that full economic model. I know many health systems, when they're deciding whether a new drug, it's certainly in the UK, you get a two-step process. You get the approval of the drug, but that doesn't mean the National Health Service is actually going to be able to afford it and prescribe it. So it then goes through a second process where they go through, how is this affordable? Surely those kind of processes that are done for new drugs, we must be able to steal a bunch of the information from that as to the affordability of home sampling. That's got to be equations that are already there for drug prescribing that can be used. I don't know. What do people think?
Is this the other thing that we should maybe be taking forward then, to see what we could do to either find tools or find out how we might go about it? Because it seems to me that if we can't get the cost benefit analysis sorted, then tackling some of the more lab-based issues downstream are of less relevance, because nobody will get agreement to take it forward. So this seems really fundamental, because it's, it's just looking at the comment that Karen put in the chat about the travel. I mean, sustainability is on everyone's agenda at the moment. So even if there was only a way that you could capture the impact on somebody not having to travel and take time off work, all those sort of aspects, or even just have a sort of value that you could nominally apply to it, to start making a case, I think, and making, finding out who we should speak to, because if this is what you do, then you would have all the answers to the questions that we've got.
Yeah, so we did a little bit of work a few years ago on when we did finger pricking, because the consumables, the plastics, there's a lot less plastic than the venous. We throw away a lot less blood, we throw away a lot less plastic. We don't use the big bio bins now, we use a much smaller one, so we've probably reduced our bin of clinical waste by about 2/3. However, the transdermal devices add in a little bit more plastic, add in a bit more complexity that can't be recycled. But like the traveling to and from clinic, the plastic wastage, the binning of the blood, all these things add into that that thing. And I think it would be a fantastic exercise to get at least some sort of idea of what it costs. I mean, I know there was a paper a couple of years ago that looked at people getting capillary blood for COVID antibodies compared to going into the clinic, but the price that they charge, I mean, it was a lot more expensive to do venous, but the price that those people had been charged was the NHS's standard charge, wasn't actually what it cost. So it wasn't quite a like for like. So that having that work done would be, well, could change. And then once you've got that, you need to go to the NHS then and say, this is the difference.
Yeah.
Yeah.
I mean, the environment, so the impact of having to people just take time off work. When we have a bank holiday, it costs a fortune. They talk about billions. So if everyone's having to go to the clinic every time to get a phlebotomy, that's got to have an economic impact. So calculating that would be fantastic.
And children taking time off school.
And for me, that's where it's so important to actually get those payers around the table to actually say what is going to drive that investment. So there's a long list, but what sits on the top 5 or top 6 or top 10, and that can actually make them to just shift that goal. That would say, yes, we can definitely invest in this, and that would be critical.
And I'm seeing people sharing a lot of potentially resources online here. And, maybe there are some of the answers are already out there, but maybe we need one of the outputs of this is a team to just look at this, to look at what information is already out there, what information is missing, and what information is needed, and do a consultation and then make a commitment to producing that kind of documentation. So to me, it's sounding like that's a good ongoing activity that might arise from that. Do people kind of agree with that?
Neil, I would agree. There is here, certainly in the United Kingdom and in each of the 4 nations of the United Kingdom, a health technology assessment and evaluation process done by certain organizations. There's an equivalent of that in the EU called the EU HTA system under certain regulation 2021-22282, which does something similar, which could be replicated on.
Okay, yeah, because if there's things already out there, we don't want to invent something that's already there. If we just need to draw people's attention to it or modify it or whatever, that's the important thing. So bear in mind we've got just 5 minutes left. Does anyone have any final comments before we draw all this together?
I was just going to say that I think it's always problematic where if you do a cost benefit, if the savings are afforded, for example, to the patient, but it costs more for the system. And that's often the case with point of care testing. and perhaps with this type of testing, microsampling, is that on a unit cost basis, it may be more expensive. This savings might be afforded to the individual who doesn't have to miss work or whatever. But you have to sort of find a way that you can convince the system that it might, worst case scenario, you could say it might keep a patient out of the hospital or something like that, looking for savings for the system.
Yeah, absolutely, Sharon, because you can see a lot of these, buying these devices and adapting tests is going to be more expensive for the lab, and therefore the lab's going to be not want to do this, even if they see the savings made in the whole system and if it's beneficial to the patient. It's complicated, but I guess we have to start. We have to try.
Yes.
So,
Neil, I just have one quick question is what's the position of Project Comfort in this, funded by the EU? Is there also potential data collected with regards to cost benefits? And
I think there will be, and we need to look at that, Vincent. A number of us are involved in Project Comfort as well, and just making sure that what we do, whatever activities we come up with, are aligned with what Project Comfort's doing as well. And we don't duplicate efforts. Likewise, with whatever may be getting done by different organizations around the world, hopefully whatever we launch as activities here is something that's not being covered elsewhere. So I'm just we're coming to the end of the time now. Thank you everyone for a great discussion. So over the next few days, we'll be compiling all the comments, because it's been a lot going on here, a lot of conversation, a lot of stuff happening through the chat that I probably haven't picked up on. I apologize if I missed your comment or whatever. We'll try and pick up on that later. We'll be taking the similar output from the events happening in North America. So their challenges event happens next Thursday. Their solutions event happens on May the 7th. Registration is open for those if people want to attend those as well. And I think what I'd like to see is we then compile this information into things that we share on our website, on the LinkedIn group. But I'm already seeing at least three groups that may potentially come out of this to follow on this with working groups around the labs, the patients, the economics that we've been talking about. And maybe there's some other things we've missed as we go through. We'll take a look at that. But if people have any further thoughts on this, please write to me and I can add it into all the discussion that's going on. So my email address is there for those that don't already have it. Also, if you would like to take part in any of the follow-up activities, please email me. Once we get a little bit more focused, after we've digested this information from this event and the North American event, we'll be putting requests onto the PCSIG LinkedIn group for people to come forward for different working groups. But I think I'm seeing at least 3 working groups coming out of this at the moment around economic use cases, around patient and around labs and engaging the manufacturers, etc. I've missed everyone. Okay, hopefully that's a little bit of a summary. So just reminding people, if you're not a member of the Patient-Centric Sampling Interest Group, it's free to join. Just hold your camera up to that QR code on the right-hand side to join the LinkedIn group. There's lots of resources on our webpage. Please use the QR code on the left-hand side if you want to take a look at that. I want to thank our sponsors again, but most importantly, I want to thank all the presenters, all the panelists today. It was a fantastic discussion. I want to thank everyone online. There's been lots and lots of discussion and communication here. my head's feeling a little bit like exploding at the moment, but that's for a good reason. It was a great conversation. The recording of this event will be available on our website in the next few days, and as I say, we'll be compiling this. This isn't where it ends. It's not what PCSIG does. We're about solutions. We're about moving things forward, not about just talking about things. We're about doing and delivering. So please, if you want to be part of that solution, please just let me know. And really looking forward to the next steps. So thank you, everyone, for a great discussion today.